I have given different lectures on cell biology, the cell organelles present in cells. Now, one of the important part of the cell is nucleus. Nucleus was first discovered by a botanist, Robert Brown. He was a botanist and he discovered it. He also gave the name nucleus to it. And the nucleus is present only in eukaryotic cells. So they are called eukaryotic cells. Due to the presence of nucleus, it can be present in plant cells, it can be present in animal cells, fungal cells. However, there are some cells like RBCs where it is absent. Now, as for the number, what will be the number of nuclei in a cell? Most of the cells, if you will see, they have only one nucleus, so they are called uninucleate, or we can call them mononucleate. Most of the cells, I have said, are having only one nucleus. We call them uninucleate or mononucleate. There are some cells, like if you remember, kingdom Protista, there is paramecium, a protozoon, have been placed in kingdom Protista. It is actually having animal origin, but later was delimited and placed in kingdom Protista. It has two nuclei, so we can call it binucleate. Paramecium, you should remember, it is a protist. Actually, it has animal origin, but placed in kingdom Protista, it has two nuclei, so binucleate. If you will take the muscle cells of human beings, muscle cells, these have many nuclei, so these are called multinucleate. Similarly, you can take a fungal cell. A fungal cell is usually called hypha. You remember it? In kingdom fungi. For example, Rhizopus is there, which is commonly called a bread mold. Its cell is somewhat long, so called hypha. It is also having many nuclei, so it can be multinucleate, sometimes called sinusoid. So, usually these are four categories of cells on the basis of number. A uninucleate or mononucleate cell with one nucleus. A binucleate cell like paramecium having two nuclei. A muscle cell having many nuclei, multinucleate, or a fungal hyphal cell having many nuclei called multinucleate. You can call it multinucleate or sinusoid. Now, as far the structure of the nucleus is concerned, nucleus is a double membranous structure. It has two membranes. Usually these are called envelopes, nuclear envelopes. This is outer nuclear envelope. And this is inner nuclear envelope. So, these are two nuclear envelopes. Both the nuclear envelopes have same structure, same constitution as of cell membrane. And I have told you in my previous lecture that cell membrane is formed of lipids and proteins. Similarly, both the envelopes are formed of lipids and proteins. So same structure. Now, there is a space between the two. This is called perinuclear space. This space. It is about 30 angstroms, 30 to 40 angstroms if we will see deeply, just like unlike this plasma membrane or mitochondria, chloroplast, I have told you, there are two membranes were somewhat different. Outer was permeable, inner was semi-permeable or selectively permeable. That means outer allowed the whole material to pass in and out, but inner membrane was selectively permeable. It actually allows that material which is to be taken inside the cell for activities or to be thrown out. But here somewhat there is difference. Both the membranes are traversed by numerous pores. Many pores are there. These are called nuclear pores. Both the membranes, a continuous pore channel called nuclear pore. Actually, its structure is such. It is usually called nuclear pore complex. Nuclear pore complex, NPC. Nuclear pore complex, NPC. Now, what is nuclear pore complex? It is that pore. It's actually a type of gate. It acts as a gate. That means it will allow the material on selective basis. For example, the material has this is the periphery, this is the cell. Material passed from cell to inner side. It will not continuously allow it. It will allow it if it is needed by the nucleus. If it is not needed, it will not allow it. Similarly, I will tell you that in the there are some RNAs, types of RNA. Few RNAs are allowed to exit, some RNAs not allowed. So this forms now it is a type of gate, so-called nuclear pore complex, and it is essential. It makes the communication between nucleus and the cell. This is cell sap or cytoplasm, and here in the nucleus there is nuclear sap. Nuclear sap, often called karyolip. It is also called karyolip. And outside, this is the outside cytoplasm, and the nucleus has to get its nutrients and other relevant material from cytoplasm, and these pores, which I have told you, nuclear pore complex, these are essential, and these are the only means where transport will occur from outside to inside or from inside to outside. Now, this is filled by nuclear sap or karyolip, which is also a mixture. And the main thing in the nuclear sap complex is it is DNA. Here, DNA is in the form of network. It is associated with special basic proteins called histone proteins. There are some non-histone proteins, and these three, DNA, histone proteins and non-histone proteins. Three, you should remember: DNA, histone proteins, and non-histone proteins. Non-histone protein. These form a type of complex, and which is called chromatin network. It is called chromatin network. Chromatin network. It is called chromatin network. Chromatin network is an important part of the nucleus. It is present in the nuclear sap. It is composed of DNA, histone proteins. Histone proteins are basic proteins, and non-histone proteins. Non-histone proteins. And this chromatin network, it controls overall activities of the cell or nucleus are controlled by this chromatin network. It varies genes. It can control features. It can help in synthesis of protein. It can control all the metabolic activities of the cell. Now it has been seen that is chromatin network. Some part of it is active, some remains non-inactive. That means some functions and some part does not function for a particular time. That part of the chromatin network which is active, it is called euchromatin. It is active. That means it is functional. It continuously works. And if you will recognize it from the other part and we are placing some basic dyes on it, it usually dyes light color. That means staining. If we stain it, if we color it, it will stain light. It will not take much dye. Now the other part of the chromatin network, it is usually becomes inactive for some time. It can be activated, but it remains inactive for some time. It is called heterochromatin. Heterochromatin. It usually takes dyes dark. That means it takes much dye, and it is somewhat thicker than euchromatin. It thickens actually. It loses some water and thickens, so it becomes somewhat thick than euchromatin. So it takes dark light. This takes light. This turns light. This turns dark. But heterochromatin does not always remain inactive. When the cell needs, it can be activated and converted into euchromatin. So this euchromatin, you are aware of chromosomes. Actually, these are also chromosomes. But DNA is there. It is associated with histone and non-histone proteins forming the complex that is called chromosome and chromatin network. Now, the main function of chromatin network is it bears genes. It helps in the synthesis of messenger RNA, and messenger RNA along with their genes, along with one more RNA, ribosomal RNA, along with one more RNA, transfer RNA, it takes the help of ribosomes and amino acids, and this helps protein formation. Protein formation is the important function of eukaryote. Now, see, genes are located on chromosomes or DNA. Messenger RNA is synthesized by DNA, and then it moves into the nuclear sac, from the nuclear sac into nuclear pores and into cytoplasm of the cell, which is here as messenger RNA. Then other RNAs, transfer RNA, ribosomal RNA, these are present already in the cell. These have been made by the these nuclear components and left the nucleus and entered the cell. Ribosomes are also present in the cytoplasm along with amino acids, and the formation of proteins occurs in the cellular cytoplasm, not in the nucleoplasm. So protein synthesis does not occur inside the nucleus, it occurs outside the nucleus in the cytoplasm. Now, ribosomes are important. For that purpose, there are some 1 to 4 bodies here. These are called nucleolus. The cell may contain 1, 2, 3, or 4 nucleolus. These nucleoli, they help in the formation of these ribosomal RNA extra along with the ribosomes, so that protein synthesis is achieved. So, as you know, the nucleus is essential for cell division. Chromosomes are actually part of the cell division or DNA. It replicates and it helps in cell division. So nucleus is the important cellular organelle, and in fact, if nucleus dies, the cell also dies. The cell cannot survive. However, there are some cells like red blood cells of the human beings or different these vertebrates. They do not contain nucleus, since I have told you that this RBC have to store hemoglobin. So in order to leave the space, nucleus and other cell organelles, they have to be removed. Otherwise, much hemoglobin cannot be stored. So this was the structure of nucleus and some of its functions. Thanks.